Hi everyone, I'm Doc Ken and welcome to Science, Technology, and Society, Chapter 6 entitled Gene Therapy. So, what are the learning outcomes for this chapter? One is to demonstrate knowledge of gene therapy and its various forms, to identify and distinguish the two types of gene therapy, and third, appreciate the importance of gene therapy. So, what is gene therapy? Gene therapy is an experimental technique that uses genes to treat or prevent disease. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery because some diseases are caused by errors or mutation in specific genes. Gene therapy delivers DNA into cells to replace mutated or missing genes or to add good genes. How does gene therapy work? Gene therapy is designed to introduce genetic material into cells that compensate for abnormal genes. Here, the patient is being given a healthy version of gene that makes the cell functional and restored. As you can see on the figure, a 9-year-old boy named Creed, who has a genetic form of blindness, is one of the first people in the United States to receive a recently FDA approved gene therapy for a congenital form of blindness called Leber congenital amaurosis. The treatment is an injection of virus delivered through a hair-thin needle underneath a patient's retina. Within a month of gene therapy, Creed was able to see details of the world he would never seen before. Let's now move on to the two types of gene therapy. Virtually all cells in the human body contain genes, making them potential targets for gene therapy. However, these cells can be divided into major categories: somatic cells, most cells of the body, and cells of the germline, eggs or sperm. In theory, it is possible to transform either somatic cell or germ cell. There are two different types of gene therapy depending on which types of cells are treated. One, germline gene therapy involves the modification of the genes inside germ cells, sperm, or ova. Two, unlike germline therapy, somatic gene therapy only involves the insertion of therapeutic DNA into body cells and not the germ cells or gametes. This means any effects of the therapy are confined to the individual being treated and are not inherited by future offspring. In summary, the distinction is the results of any somatic gene therapy are restricted to the actual patients and are not passed on to his or her children. All gene therapy to date on humans has been directed at somatic cells, whereas germline engineering in humans remain controversial and prohibited in, for instance, the European Union. Let's now move on to the two types of somatic gene therapy. In vivo gene therapy. In vivo gene therapy means the therapy is administered directly into the patient. The targeted cells remain in the body of the patient. The gene is being transferred to a patient's cells while still in the patient. However, a gene cannot be introduced directly into a patient's cells. Instead, a vector, which refers to a virus, is genetically engineered to transport the gene. Question: will the virus make a human sick? The answer is no. Once the virus delivers the gene into the cell, the virus slips away. Certain viruses are often used as vectors because they can deliver the new gene by injecting the cell. The viruses are modified so they can't cause disease when used in people. Next type of somatic gene therapy, ex vivo gene therapy. In ex vivo gene therapy, the targeted cells are removed from the patient and gene therapy is administered to the cells in vitro before they are returned to the patient's body. Here, the gene is being transferred into the cells in laboratory and then injected back into the patients via IV directly into a specific tissue. So as you can see in the figure, in ex vivo gene therapy, the cells are being taken out from the patient and these cells will be modified in the laboratory. When the cells become restored or functional, the cells will be now returned into the patient's body. Let's now move on to the recent developments of gene therapy. Researchers are testing several approaches to gene therapy, including one, replacing a mutated gene that causes disease with a healthy copy of the gene. Two, inactivating or knocking out a mutated gene that is functioning improperly. And third, introducing a gene or a new gene into the body to help fight a disease. Although gene therapy is a promising treatment option for a number of diseases, including inherited disorders or some type of cancer and certain viral infections, the technique remains risky and still under the study to make sure that it will be safe and effective. Gene therapy is currently being tested only for diseases that have no other cures.